Hello students, I am Akshay Deshpande and welcome to biology class. And in this class today we will be beginning a completely new chapter that is biomolecules. And biomolecules is fairly an important chapter. I can say this chapter is a lot of biochemistry. Okay, this is mainly a kind of biochemistry chapter where the intention of the whole chapter is mainly to introduce about the molecules that are involved in the living system and what are their purpose and what do they do in the living systems. That is what the main purpose of the biomolecules is. And this is the first class, as you people all can see over here. And today we will be starting with the first part of the chapter, that is how to analyze any chemical composition in the living systems. Right? So let's get started then. Now, if you look into our planet and if you take any living organism, okay, what we mean by over here is any tissue, okay, any tissue if you try to take from any living organism, and also if you try to analyze the contents of the earth crust, okay, mainly with respect to we can say the soil samples, right? If we take soil samples, right, and if we analyze it with respect to by comparing the contents of the living tissue and also by comparing the contents of the soil samples, you will see that there is no much difference with respect to the constituents of the tissue and also the constituents of the earth crust, right? For example, the major of the contents that you will see over here are the carbon, the oxygen, the next one we are having is the hydrogen, and finally some other similar lists, right? So we can say that we are no different than any other constituent of the planet Earth. Ultimately, we are made up of all the basic compounds or the minerals, we can say, that is carbon, oxygen, hydrogen, and some other inorganic constituents. So this is mainly about the comparison of the living systems and also the earth crust. And here we are actually having a table which shows the different elements, okay, different type of elements, and also the percentage of weight of the earth's crust of these constituents and also in the human body. Right now I have mentioned the table directly from the textbook, and I have seen that some of the questions do have appeared from this particular table, so we can say that this table is quite important for our discussion, right? Yes. The first element we can see over here is the hydrogen, and the percentage in the case of earth crust is around 0.14 percentage, and in the case of a human body it is only 0.5 percentage. On the contrary, you can see that the carbon is around only 0.03 percentage in the earth crust, but whereas in the case of living tissues of human body, you can see it is around 18.5 percentage. And also in the case of oxygen, you can see that it is one of the most abundant element present in the earth crust, that is around 46.6, and in the case of human body it is around 65 percentage, right? So if you look into all other constituents, you can easily notice that the oxygen is very important in the terms of the percentage of weight in the earth crust and also in the human body, right? So in the earth crust the most abundant is the oxygen, and even in the case of human body it is the abundant. But when you compare between the human body and also the earth crust, you can clearly see that oxygen is more in the human body than even in the case of the earth crust, right? The nitrogen is very little over here, right, in the case of earth crust, but whereas in the human body it is around 3.3 percentage, and sulfur is around 0.03, same as the carbon, and 0.3 in the case of human body, and sodium is around 2.8 percentage in earth crust and 0.2 in human body, and calcium we are having 3.6 and 1.5 in the case of human body, and magnesium is 2.1 and 0.1 in the case of the human body. Silicon we are having around 27.7 percentage in earth crust, but however the same element in the human body is completely negligible. right? So now they have mentioned all of these elements, okay? And if we have to remember the important ones, okay, if you have to emphasize the most important ones from all these mentioned elements, we can say that try to remember the major constituents, major elements, okay, major element percentage, right? See, if you consider the earth crust, you can clearly see that oxygen is the major. The next comes we are having is the silicon, right? And the next one we are having is the calcium, right? So try to remember the first top three major constituents in the earth crust, okay? And also try to remember the least values, okay? So if you consider the least value, we can say that the nitrogen is completely very little, okay? You can try to remember this. And carbon, and finally we are having is sulfur. These three are the least, okay? So highest we are having is the oxygen, and silicon is highest, and finally we are having that is the calcium being the next highest, right? The top three. But the nitrogen is the least, okay? And also the carbon is the least in the earth crust, and also we can say that the sulfur is also least, right? The up arrow mark indicates the highest, and the down arrow mark mainly indicates the lowest one. And similarly, if you compare with the human body, try to remember the highest values. As you people can clearly see, that carbon, oxygen, and the next one we are having is the nitrogen, the three clear winners for our remembering, right? The first one we are having is the carbon. The highest quantity we are having the oxygen, the second we are having is carbon, and the next highest we are having is the nitrogen, okay? And also now in the case of human body, if you try to recall or try to remember the least constituents, you can see that silicon is the least constituent. Next comes we are having is the magnesium, and the next one we are having is the sodium, right? So try to remember these three highest and these three lowest, okay? So that is how the questions are generally framed, that they will mainly concentrate on the major elements and also the minor constituents, okay? So you don't have to worry about the intermediate values. Nobody is going to ask you about the intermediate values, okay? And in this way you can just try to make a small table for your reference and try to recall them every day as much as possible, okay? Ultimately this we have to try to make a chart, make a concept map, and recall them as much as possible, okay? That is how you can make a firm confirm memory regarding this, okay? We have seen, let me tell you, like I have seen many questions where they have asked which of the following is a minor constituent or a major constituent or completely negligible or very little. They have asked this, okay? So such kind of questions are direct questions which will yield you 4 marks, right? So try to, as I said, try to make a note on these list of elements that I have mentioned in your textbook. Now let us go on to the next one. That is organic compounds in the case of organisms, right? So now how can we determine the organic constituents in the tissues of the living organisms? It is quite a simple task if you ask, because we can take a tissue. If you want, you can take plant. It is very easily accessible and even in some cases you can even take the animal tissues also, right? And we have to grind this particular tissue that we have taken with a chemical called as the trichloroacetic acid, okay? We have to grind it with the trichloroacetic acid, right? So they have mentioned the formula Cl3CCOOH. This is the chemical formula for the trichloroacetic acid, right? So you have to grind it in the mortar and pestle, okay? Mortar and pestle, right? If you grind it in this equipment, you will obtain a thick paste like a semi-solid slurry, okay? And we have to strain it, okay? We have to strain it. That is straining is a general process that you people have seen in your household, right? We have to take a cloth and place that cloth in a bowl or a container and put that particular slurry in this cloth, that is a muslin cloth, and what happens is that you will land up with the liquid substance, and also you will land up with the solid substance, right? So now this filtrate we are very much concentrating, right? We are getting mainly two types of fractions. The first one we call it as the acid soluble fraction, which is mainly present in the filtrate. Okay, this acid soluble is mainly present in the filtrate, but whereas the acid insoluble is mainly present in this the cloth leaving what we get. So the acid insoluble and acid soluble, why they are called as in that way? Because the type of organic solvent we are using over here, that is the trichloroacetic acid, right? So all the organic molecules which are present, they will be present in the filtrate, and the filtrate is mainly made up of the trichloroacetic acid, right? So now we can actually determine the constituents in the acid soluble fraction. Okay, and we are having a technique called as the chromatography, right? So chromatography, I hope you people can remember from your first year practicals. There's a demonstration called as the paper chromatography, right? We are mainly using the Whatman filter paper, and we use an organic solvent in a cylinder or a big jar, and we will dip our chromatographic paper in the jar in such a way that only a tiny tip of that particular paper touches the buffer or the organic solvent, right? So due to the capillary effect, what happens, the solvent will actually rise through the paper, and the paper that is containing the mixture of solvents or the substances will gradually separate based upon the affinity. Those substances will show towards the organic solvent that we are using, right? So hence we can say the chromatography is used for the purification and separation, right? There are many other techniques. For example, we are having centrifugation, right? It is mainly used for separation but not for purification, right? And also we are having electrophoresis. It is also used for separation but not for purification. Remember, both with separation and purification, we are having a technique called as the chromatography, which is widely employed in various laboratories. So now, using these technique, that is mainly the chromatography, we obtain the molecules which are present in the living systems called as the biomolecules. And in a simple way, in the most briefest way to define a biomolecule, we can just simply call them as a carbon-derived molecules. We can say that it is mainly a carbon-derived molecules, right? So now these are the carbon-derived molecules. But what about the inorganic nutrients? Inorganic nutrients. Can we identify the inorganic nutrients? Yes, we can identify the inorganic nutrients, but we have to do a destructive experiment for this. Destructive experiment. Right? So what do we mean by a destructive experiment? It means that we have to take the tissue, dry the tissue, and after drying the tissue, we have to burn it. Right? So the intention of drying is mainly to remove the water content in the tissue. Right? Hence we are drying it and we have to burn it, and finally the remains of the tissue is called as the ash. Right? And if you analyze the ash contents, you mainly find inorganic nutrients. For example, calcium. For example, we are having magnesium, okay, and sodium, right? So on. All of these we can say that inorganic nutrients, and interestingly, even these inorganic nutrients are also coming under the fraction called as the acid soluble fraction. Yes, acid soluble fraction. So now we are having ultimately organic constituents, we are having inorganic constituents, and also we are having the functional groups, right? Now, for example, the functional groups, we are mainly using it for the classification. Okay, we mainly try to determine the functional groups, okay, from the chemistry point of view. From the chemistry point of view, right? So here we are mainly concerned with the functional groups, right? But in the case of biological point of view, from the biology point of view, we can say that we just classify the biomolecules, and that is whether they are amino acids, right? Whether they are amino acids or they may be carbohydrates, right? They may be carbohydrates, right? Or they may be proteins, and etc. Right? So simple classification, we mainly discuss it from the biological point of view, and if you are looking into the functional groups, generally we discuss it from the chemistry or chemical property of the substances or the molecules.